Hi, I'd like to talk to you today about the risk of viral transmission from shedding and processing fish. Viral hemorrhagic septicemia virus can cause severe disease and major economic losses in aquaculture facilities, mainly in the salmonid industry. The UK is VHSV disease-free and the disease is notifiable to the OIE. We've previously found that preclinical tissues of infected fish can contain high levels of virus, but until a disease is recognised, processing continues to take place. EU legislation minimises the risk of notifiable disease transmission by restricting movements of live fish depending on a country's disease status. However, trade of eviscerated fish carcasses imported into the EU or traded within is not restricted. So as a result, commodities can be imported into the EU and other countries applying the same principles, including the UK, carrying exotic pathogens. Potential risk of disease introduction is from the wastewater produced from processing these fish. As such, the aim of our study was to quantify the amount of virus shed from infected fish and the amount of virus released in the wastewater from processing preclinical fish. Daily water samples were taken to quantify virus shed from infected rainbow trout. Preclinical fish were gutted and the same fish filleted, during which the wastewater was collected. Half of the samples were blended to mimic potential maceration at a processing facility. Virus was quantified in the solid and liquid fractions of the wastewater by cell culture titration and compared to the quantities in the kidney and skin and muscle of the same fish. First detection and peak shedding was actually before clinical signs of disease. Average levels in the wastewater were relatively high from both processes, high enough to potentially cause an epidemic. It's important to remember that there are many factors influencing transmission and infection. These could be mechanical, environmental, chemical, or locational. However, the quantities of VHSV from shedding and processing can be utilised when modelling transmission and undertaking more accurate risk assessments for imports and processing of commodities. This evidence, further data, risk assessments and modelling will be used to inform policy and legislation, with the ultimate aim to reduce the global risk of disease.